Good afternoon, everybody, and welcome to UTS Science in Focus, which is a public free lecture series showcasing the latest research from prominent UTS scientists and researchers. I'm Professor Bradley Williams, Associate Dean, External and International Engagement, and I'll be the moderator for today's event. Part of my role is to collate the audience's questions and pose them to our speakers at the end of the talks. Our Science in Focus talks provide an outstanding platform for UTS academics and public participants to meet for the sharing of ideas and to shine a spotlight on some of the top-of-the-line work we're doing at the Faculty of Science at UTS. Not only are we pushing the boundaries of scientific knowledge by making new and substantial contributions to a fundamental understanding of the world around us, but we have a dedication and devotion to translate our work into real-world impact. Today, we will hear from one of our UTS experts making major contributions to our knowledge and understanding of newer and easy-to-use tests for SARS-CoV-2, the virus causing COVID-19. We will also learn about the rollercoaster ride of one of Australia's largest private pathology companies and how COVID affected them too. Before I introduce our speakers for today, I'd like to acknowledge on behalf of everyone here present, the Gadigal people of the Eora Nation, upon whose ancestral lands the UTS City campus now stands. I would also like to pay respect to the elders, both past and present, acknowledging them as the traditional custodians of knowledge for this land. I'd also like to recognize the Camaragal people of the Ku-ring-gai Nation from which I join today, and the people and elders of the country from which you join today. Just a little housekeeping, and I will forward this slide before we get started. It is an online event, and there have been some technical difficulties with internet connectivity today, so if there are problems, please bear with us and we will resolve them as quickly as possible. If you have any questions during today's webinar, please use the Q&A function in the Zoom, and you'll find it as shown on the screen now. And you can then enter your question into that Q&A function. If you like a question that you've seen, you can always use the upvoting tool, which is the little thumbs up symbol next to the question itself. This session will be recorded, and it is recorded for our internal purposes, and it will be made available on the UTS website. We will not be recording any video or audio input from the audience, and you may contact us at the email address presented on this slide if you have any concerns or questions about the recording. Our speakers today are Professor Dayong Jin of the School of Mathematical and Physical Sciences at UTS, and Mr. Anthony Fridley, who's the Chief Operating Officer and Chief Executive Officer of Australian Clinical Labs. And I will introduce now Professor Dayong Jin. Distinguished Professor Dayong Jin is an award-winning scientist and technology developer who is driving the transformation of photonics and materials into analytical, diagnostic, and imaging devices for disease detection through interdisciplinary research teams. These devices identify early signs of disease and toxins to enable rapid detection of target cells and of specific molecules. He has won many prizes in recognition for his outstanding work, including the Australian Museum Eureka Prize and the Australian Prime Minister's Prize for the Physical Scientist of the Year. Dayong, I now invite you to give us your presentation. Thank you.
So thank you for the nice introduction, and thank you for all the participants and audience today for attending my lecture today. As you may notice, the title of my lecture called Part 2. Why is Part 2? Because at the end of last year, I presented on the same topic at the New South Wales Chief Scientist Breakfast Seminar Series. So on that lecture, I presented the basics of this technology, and because the time limit today, in my lecture today, I'll only focusing on the updated part. So you're welcome to visit the New South Wales Chief Scientist website and to watch my first presentation on this similar topic. So the topic today, as I said, I'll pretty much will be focusing on the new variants, and it's challenging in terms of demanding the increasing number of the herd immunity and the vaccination rate. And the focus today will be pretty much on the test, on the rapid test. There's a dilemma here between the detection speed and detection sensitivity. So I'll show you the principle, how actually each of the tests works. And then I'll bring in you to the basic principle, how our UTS science solution works and how to make the signal can be optically amplified, and in terms of the developing of a single molecule probe, which can be sensitive enough to detecting a single molecule from the virus. At the close of my lecture, I will discuss the broad range of opportunities, including the manufacture opportunity in Australia and the development of rapid and non-invasive saliva test. And of course, there are challenges ahead associated with the new variants and also the clinical translation and TJ approval process. So as I already presented a very similar picture last lecture, but this one is more simplified version to show you, how difficult to handle, to cope with this virus. At the beginning of the pandemic, I remember a lot of public, they got confused between the difference, between the bacteria and the virus. If you compare the size, the bacteria is at least 1 order of magnitude larger than the virus in diameter. But if you compare the volume, you can see the volume, the virus is 1,000 times, at least 1,000 times smaller than the size of the bacteria in volume, okay? So this COVID SARS-2, they are tiny, they are around 100 nanometer, smaller than the optical diffraction limit. So they are very, very difficult to detect. The size and the shape is pretty much closer to the size of the traditional influenza and also the HIV virus. And that's the first challenge why this is so difficult to detect. Most importantly, this virus has a very long period of incubation time inside the host people, people got infected. In the beginning of the infection, because the number of virus load is still small enough, so in the first couple of days, we call latent period, the host person doesn't pass on the virus to the other people because the number of viruses is very low. But this virus, they can duplicate themselves very quickly inside our human cell at a rate about 3 order of magnitude more virus in every 10 hours. So in the later couple of days, the host, the people who get infected will become infectious. But there's a long window. This window we call it incubation window. The time between the onset of the infectiousness and the time between, and also the time till the patient start to show the symptom. So this window is fairly long, actually can last for several days and up to 2 weeks. And this is the window is pretty dangerous because the host people has no idea whether he or she got infected and where and when they got infected, but they already passing on the virus to people surround him or her. Once the patient start to show the symptom, we traditionally using this conventional gold standard technology called PCR. But you understand you only getting the results within a day or 2. So this has become quite slow. So the entire window for understanding and detecting this virus is fairly long, can take about a couple of weeks. Okay, so this is a very, very challenging problem for this virus. And because this is a long black window, we got no idea, who is the host, who got infected. Okay, and particularly when the number, the COVID case number above 100, this is the particular true in the current pandemic lockdown in Sydney, in New South Wales. At the beginning, even at around 100 new cases, daily new cases, there are about at least half of the people who have no idea when he got infected, and they still spreading the virus in the community. But nowadays, today I heard the number is above 900 cases. So more than 80 to 90% of the patient, they have no idea. We couldn't trace these people. So because the cases they never get linked. So this is a completely annoying problem to control the spreading of this virus. And the next challenge is, the virus getting smarter. They genetically, improve themselves and they become smarter. And you can see, from the original strand to the alpha, beta, and the gamma. Nowadays, we are handling, we are coping with the Delta variant, and they are smarter and smarter. So you can see each wave, at least we are in the process of the third wave. And you can see, each wave, they could become stronger and stronger. And because they are smarter and the infection, they become more contagious. And there's a scientific number called reproductive number. It's a parameter to measure how infectious each new strand virus they are. So at the original strand, you can see the R close to 1.1. That means if somebody got infected, they can pass on by average to only one person. Okay, so the spreading rate is slower, right? But once we move to the alpha phase, the alpha variants, the number close to 2. So that means when people get infected, can quickly pass on 2 to 4, 4 to 8. So this is the power of 2, and that means very quick. And now why the Delta variant is so contagious? Because the R number is close to 3.4. So that means if one people get infected, at least 3 people by average will be infected, getting the virus passed on. And then 3 to 9, 9 to 27, and that will be quickly spreading through the whole community. So this is a pretty dangerous situation we are facing. And accordingly, in the beginning of the pandemic, we are expecting the vaccine, and now the vaccine is ready, and we predicting if 60% of the population getting the 2 shots of the jabs, and we could develop the herd immunity, so we could control the spreading of the virus within our community. But now everything changed because the new variants, and you can see the higher number of R value getting associated with the new variants, and the requirement for the vaccination rate is getting higher and higher. At this stage, we need at least 80% of the population get vaccination until we could stop the spreading of the virus through our community. And this is a new update I got from the website, and you can see at this stage, unfortunately, Australia, we been leave behind compared to the other major economy countries, including the Canada, US, European countries, and Asian countries. So hopefully there will be some good news in the next 2 to 3 months time for our population get at least 80% of the vaccination by 2 jabs. So this is unfortunately, this is a reality. And I'm sorry to sharing all of this shocking and bad news to start with my lecture. But there's some good news, okay? So this is a good news. One, the virus can be killed easily. So how the virus can be killed? By washing our hand. Because if we are using the soap molecule, they have the hydrophilic head and hydrophobic tail. The hydrophobic tail can insert onto the membrane of this virus. So the virus can be broken down into molecules. Okay, so this is the way, that's why we encourage our population to regularly washing our hands each time by soap for 20 seconds, at least 20 seconds. So once the virus break down into molecules, we have a way to detect them. And when you look at the detail of the construction of this virus, you can quickly understand, what's the, where is the strategy? How can we detect them? So there are basically two strategies and two approaches. One is to detecting the oligomolecules, which is the RNA molecule inside the virus. Each virus only have one RNA molecule, okay? The second approach is you can detecting the proteins. So we call antigens, okay? At least the spike protein, they are very specific to the SARS-CoV-2, and nuclear proteins, they are specific to the coronavirus. Okay, so then you can focus our detection to detecting these proteins. At least you have 26 copies of the spike proteins per virus, and you have 35 copies of nuclear proteins from one single virus. And that's how, where we get the detection started. PCR technology, these days is a very well known and is a gold standard, because the PCR technology can amplify the number of the RNA molecules. So from 1 to 2 to 4, 4 to 8, until you are getting 30 to 40 cycles, you're getting these target molecules amplified significantly until you get a trillion of molecules. You can detect them in the lab. But the downside, although this is a very sensitive technology, the downside we all know, this is very slow. Okay, you have to operate this in the pathology labs for a couple of hours until you got the results, which typically require overnight, 1 day to get the results. Okay, so that's explain why PCR is so sensitive, but that's also slow. There's another, sort of a good news from Channel 7 and also the public domain. You can see I'm sharing this short clip, video clip from
COVID test
Channel
results
7.
in just 15 minutes. That's the promise as new rapid testing kits are rolled out across the state. But they're not without their downsides.
It looks kind of like a pregnancy test, but this plastic stick is to be the bearer of some other good or bad news.
It's very simple to use, but the technology inside is quite sophisticated. So the tests can be run almost anywhere.
We know that it's not just the only solution, but it's part of our solution in fighting COVID.
Tom Hartley, 7 News.
So, this is some video from the news media, and also I got this screenshot yesterday from the Sydney Morning Herald website. Australia, has been fairly slow compared to other nations to implement the rapid antigen test, but it's all changing in the face of the COVID Delta variant. But there's a reason, why we're being so slow. Not because we don't know this technology, but we have a concern around this technology. I get this message, this testimony from the Twitter website. Adrian's daughter unfortunately got the COVID-19 virus recently, and he shared the experience by testing using this home-based test to monitor the positive and sensitivity of this commercial kits. And he found the lateral flow antigen test were still negative until 4 days into her symptoms. So that means, in the first couple of days, the antigen test still not so accurate. We are getting some samples, commercial kit sample from the market, and we tested in our lab. We're getting the known concentration of the protein antigen, and we check the sensitivity. And this slide shows you the principle, the animation shows you the principle, how this technology works. So at the present of this virus, you can see a virus protein, the fluorescent molecule, like a fluorescent nanoparticle, can sandwich the antigen in the middle at the testing line area. So if there are about 10 millions of the virus in the sample, the testing line start to show positive signal. So this is the current reality about the antigen test using the European technology. It's quick, only 5 minutes you can get results, but you need to have the high concentration of the virus in the sample until you can detect anything positive. So this is some limitation. Our government, the TGA, approved the COVID-19 antigen test recently, but has emphasized, saying they are less accurate. They can only be used for symptomatic patients, And if you got negative results, you still need a PCR as the confirmation test. So no matter whether you got positive or negative, you still need the PCR test. So you can see this, the reality. So if we come back to this chart, you will understand why the antigen current antigen test is still not ideal. So we got this black window, but this is where the antigen test sits. So they are not sensitive enough. And that's why we couldn't apply them to screen the people in early stages, okay? The scientific challenge for this technology is if you want to detect the proteins, the number of the protein cannot be amplified, okay? So at the beginning of the pandemic, the researchers and the scientists and also our team at UTS Institute for Biomedical Materials and Devices, we work together to consider, translating our existing technology to cope with this very difficult situation from the science perspective. And our test is basically built on this new science discovery we made for the last couple of years, and it's called nanoscopic upconversion nanocrystal. So this material is very fascinating because you could combine the multiple low energy, longer wavelength emissions and convert them into the visible emissions. So you could put a few thousands of emitters per small nanocrystal. So we're using, in this case, about 40 nanometer crystals. You can put a few thousand emitters. So that means you could first detecting the signal free of the background because you use the near infrared excitation. Second, you can optically amplify the signal by the number of emitters per nanocrystal so that you can amplify the number of the target analytes. Okay, so the journey is actually a long journey, but I cut this long story into short. We made this fundamental discovery in science about 8 years ago, and we published these results together with Professor Tanya Monroe at the University of Adelaide. We demonstrate this single crystal is sensitive enough to improve the limit of detection by 3 order magnitude compared with the quantum dot technology. We could detect a single crystal using a piece of fiber. And we further studied this technology under a microscope. We found each single crystal can emit a few thousands of photons per 100 milliseconds. So our naked eye can see the crystal under a microscope together with the cell. So you can see the transport moving in and out of a single crystal inside and outside of the single cell. So they are single molecule probes. And in parallel, a couple of years ago, we worked with Myonomic International in Sydney, a cancer biomarker company. So we translate this technology for very sensitive detection of the prostate cancer biomarkers in urine. So my student, Dr. Hao He, did a PhD with us a couple of years ago. He engineered this little box so we could attach this box into in front of the smartphone, and we could use this new lateral flow assay test to simultaneously detecting 2 proteins on 1 strip. And since the pandemic, we retailored the reagents, and now we targeting 1 spike protein, by using 1 color and targeting the nuclear protein using the other color of the probe. So we could simultaneously detecting the presence of the virus protein at the same test. So this is our latest results. We've been collaborating with researchers at the Sydney Centenary Institute and also the clinical doctors at the Prince Wales Hospital by testing our technology using the inactivated COVID virus. And we found our technology now is sensitive enough to detect the presence of about thousands to 10,000 viral particles from 100 microliter sample. And this means we already push the limit of detection by at least another 2 order magnitude. So once you push the limit detection by 2 order magnitude, and you can see from this chart, you can extend your detectable window, goes into this dark undetectable window period. So we could use this technology for detecting the asymptomatic patient. Okay, so this is of course, there's quite a large scope for us to further improve the technology until at a single molecule level. Another good news is our industry collaborator Alcolyzer. They've been funded by some federal government funding, and they retailed their fabrication labs and established the fabrication facility at Perth, and they combine the UTS technology with Alcolyzer's hardware technology, and we start to do this manufacture and commercialization process. So this is some good news. The other possibility is really started by a problem like the non-invasive testing potentials. So last weekend, I was approached by Martin. He said, he want to have a test, but he couldn't use any existing test in the testing center because they need a nasal swab, and he got some allergy issues. He couldn't use this swab-based test. He was asking whether our technology can be used. My answer was, I think this is the response to a lot of questions during my lecture. So I provided the answer beforehand. A technology works in the lab, okay? But in order to push this technology in Australian market, I think there are still quite a few steps, between the lab-based technology and the commercial product. Because the procedure to get through this through the clinical stages and the government approvals are a lot more complicated and sophisticated than we initially thought. Okay, and if when we further pushing down this limited detection by order of magnitude, we found our technology could provide a potential to do this detection from the saliva. But the big challenge from the saliva detection is, you can see from this clinical data, there's a big variation between the number of the virus load. Means, sometimes the patient who got infected can provide a very high amount of virus in their sample, like 100 million. But sometimes they can only provide about a few thousands of viral particles. So that means we need to continuously pushing the limit detection from nanogram per mil, which is a current antigen test can do, down to picogram per mil, which is our technology can do now, and further down to tens of femtogram per mil, until we could completely capture all the possibilities from the patient sample. And of course, this is a moving target. Once you start this research direction, you found more. You get more solutions. You get a lot of good excitement, good solutions, but you found more problems. And it's a moving target because the virus, they become smarter, and each step they modify their protein on the surface a little bit. Then you have to capture the latest development of the antibody technology, like a lock and a key relationship. So you have to adopt new reagents and make sure the technology can detect the new variant strand. And this is the chart to show you our current plan to push the technology from the lab-based to clinical-based and to the market and through the government approval. We already completed all the preclinical tests and showing very positive results. And now we are requesting to develop more partnerships with clinical labs and doctors to access the real patient sample. First of all, we need maybe 20 confirmed patient sample to start to understand the variation, how much virus we could detect from confirmed cases. Then we upscale into 200 confirmed patients in the mix of the healthy patients to check the sensitivity and specificity. Then we could use this technology to unknown cases, like a larger population of unknown cases. Once you complete all this data set, you can provide all the documents to the government and for the CE, FDA, and TGA approvals. Okay, finally, I would like to thank my colleagues and my postdocs and the students. They are extremely efficient and hardworking members at IBMD. Particular During the pandemic, they are still active and productive in the lab. They sacrifice a lot, these days, and try to push this, accelerate, the development of the technology in the lab. I sincerely, appreciate their efforts. As I said, this is a joint effort between different groups and between, UTS and also industry partners and external partners from hospital. And of course, without funding, we couldn't do this research. I would like to acknowledge Australian Research Council for continuous funding our research from the fundamental discovery level to engineering level, now to the translation towards the industry usage. In particular, we would like to thank the Innovative Manufacture Cooperative Research Center for directly funding this research towards the commercialization and industry prototype development. Thank you, everybody.
Thank you very much, Dayong. I really appreciate your talk today, and also for highlighting some of the issues that we face. In particular, we in New South Wales every day hear from the Premier, and we hear the numbers of people infectious in the community. And of course, you've now explained why that is the case, even with the very best of testing. And there's certainly hope for these quicker tests, as you've identified, which will hopefully bring back that window to enable much quicker testing of people who are pre-symptomatic or early symptomatic. So yeah, thank you very much for that. I'm now going to introduce Anthony Friedley, Mr. Anthony Friedley from Australian Clinical Labs. And before I do so, I suppose the main point that I'd like to make is that notwithstanding any of these quick tests when they come through, when they are positive, of course, we still need to submit the patient testing for testing at a PCR lab, such as what Anthony will describe shortly. Of course, this is really important for the contact tracing and for the strain detection. So it's only really in the labs under those with the PCR technology that Dayong referred to, where we can achieve the type of tracing and strain detection. So let me introduce Anthony. Thank you very much, Anthony, for joining us today. Anthony Friedley is Chief Operating Officer and Victoria CEO of Australian Clinical Labs, and his responsibilities include leading the business transformation on a national basis and managing in toto the Victorian operation. Anthony also oversees human resources, quality and risk, and business improvement functions right across Australian Clinical Labs. Prior to joining ACL, Anthony was Managing Director of Australia and New Zealand for Kepner Trego, a management consulting company specializing in business transformation. And Anthony has also held management roles within the telecommunications, banking, and manufacturing industries. Anthony, thank you so much for joining us today, and I invite you to give us your presentation.
Many people start a career with a particular end game in mind. Some people keep to that path, while others go off on tangents and explore new frontiers. Many of you have heard of some of those success stories of careers that seem to evolve rather than follow a plan or a path that's well trodden. I believe your degree not only it does not define you, it actually enables you to do amazing things. Today, I'm going to take you on a journey of how a UTS engineering graduate found himself managing at the front line of a global pandemic. I will talk about the roller coaster ride that is pathology, has been in the past year, as well as a prediction into the future of what the costs will be and what issues and potential health risks are there within the community. So back in 1996, I graduated with a bachelor's degree in electrical engineering, majoring in telecommunications. Throughout my time at UTS, I had a cadetship with Westinghouse and worked my way through that organisation and eventually leaving and taking a role up in one of Australia's largest telecommunications companies, which was Optus. At this point, you suggest my logical career path had been achieved. What else would a telecommunications professional end up. So and then in 2015, my career was provided a tangent, and I found myself as the Chief Operating Officer and Victorian CEO of Australian Clinical Labs, a newly formed pathology company amalgamated from Healthscope Pathology, St John of Pathology, and a number of small players. An electrical engineer now running a pathology company. Really, pathology is a tough business. Only the top 2 private pathology companies actually make any money. All the rest run at a loss. One payer, which is the government, 20 years of no fee increases on top of 3 major fee cuts means the industry is on its knees. I was brought in to salvage a number of companies, and 4 years later, we managed to save these businesses from extinction and prevent the country from having another duopoly. The month was February 2020. My CEO calls me into her office and proclaims that we have successfully saved the organisation from closure, and we will celebrate in March. We knew about coronavirus since November 2019 and had proactively worked up a test that would be able to be used if required. Little did we know what was about to happen. I'd now like to take you on a journey of what COVID-19 looked like from the viewpoint of a pathology company. The way I will portray this journey is through the lens of work coming into our organisation. The axis on the left shows the percentage comparison of volume from previous years. The dark line across the graph is the 0% line, which means that you're not making any money. Above the line, you're okay. Below the line, you're losing. So what I have done is broken this story down into 7 parts. February 2020, after 4 long years, the business was making a very meagre profit. It was enough to be invested back into the organisation, and that organisation now could be sustained. 23rd of March 2020, disbelief. As our country was overcome by the fear of COVID and we plunged ourselves into lockdown, we watched as our organisation collapsed. To give you relativity and to use a boating analogy, our business at minus 10% on this graph starts to take on water. But at minus 40%, we've hit an iceberg. Work dries up coming into the lab. Patients abandon their doctors. Private hospitals are ordered to close. Public hospitals are empty to make space for the anticipated wave of COVID patients that we saw in the US and UK. The world is confused and it stops, and so does our business. A quick calculation reveals that we have 45 days before we will need to close the doors. We will be out of business by mid-May. 30th of April 2020, we are drowning. With work barely trickling in, we have no way of paying our people. The government steps in and announces a JobKeeper program for businesses that have slumped to over 30%. We hurriedly apply, and within 2 weeks, we have enough money to pay our people. Not the full amount, but something. We decide not to lose any staff, but reduce all pay. This lifeline does not save us, but it gives us another 20 days to survive. It's a dim light at the end of the tunnel, but it's a light nonetheless. June 30, 2020, gasping for air. With pay rates in some cases down 80% and staff stood down all over the country, COVID testing starts to ramp up considerably. Then a new threat hits our organisation. One department is now functioning at levels never seen, while the other 9 scamper for work. As we retrain scientists, we realise the biggest threat to our organisation and our existence is now our supply chain. Issues we now face. 1, PPE to manage the collection of COVID is now in short supply, and we are connected to a secret department in Canberra for supply. The reagent for our equipment can't be produced fast enough, and we're a small market in Australia, so our allotment is difficult. We strategically decide not to partner with one supplier, but to deal with 4. The variation is tough on our people, but keeping supply is key. Qualified staff become tough and in short supply. With borders closed, it is tough to find personnel willing to work the grueling 24/7 shifts that now keep up with demand. Swabs become impossible to find, and all sorts of fly-by-nighters have popped up trying to make a profit in this dire circumstances. Again, the federal government steps in and provides us with critical supply. Masks now become difficult to source and therefore slow down our operation. Our lab staff are allocated masks, but if we miss one shipment, we will have to stop the testing, and that will put pressure on our time, on the turnaround times back to the public. So we are walking on a tightrope. The 9th of August 2020, resilience. As New South Wales, South Australia, WA and Queensland, as shown in the green line, start operating in a COVID normal state, Victoria and the hotel quarantine bundle sends our business into a spiral. Our organisation is heavily skewed to Victoria, and healthcare again plummets in this state, dangerously low levels, with hospitals and doctors' surgeries closed. We've been here before, and we've got good processes, good supply chains, and the testing we're completing keeps us afloat. But what will be the true effect of this second lockdown? 15th of October 2020, hope. Our organisation is almost back to pre-COVID levels. We are expecting a surge of testing from the consults that never happened over the past few months, but those never come. Our standard tests return and hospitals come back online. Theatre lists fill up and elective surgery starts to come back. COVID testing is now constant, and as the experts believe that it's here to stay. Our profits return and we repay our JobKeeper payments. Our business has survived, but what is the toll that it's taken on our people? December 2020. How do you manage in this environment? Pathology is now in the limelight. We are weary that we've not only survived, but that we've thrived. We now have new technology and aids to speed up testing. We have processes and reporting that can navigate through the toughest situations. We have formed relationships and bonds that will help us into the future. The threat of the next outbreak is never far, but we are ready. We are ready and capable. This story has continued throughout this year, from Victoria moving into its 6th lockdown to New South Wales now suffering through the longest lockdown ever. I hope that this timeline has dispelled some of the myths of the affluence and good times that pathology must have been feeling. I can honestly say that these were the scariest times of my life. This story has much more to unfold. What does the future hold? How long will COVID-19 testing be a thing? What does antibody testing look like? COVID-19 passports, what are they and how do they work? Next winter, how long is the vaccine going to be effective for? New strains, new severity, new challenges. Will we ever travel again? And how will it look? Pathology has been a driving force in the management of the pandemic. But what have been the costs? Dr. David Dean and Dr. Simon Nazaritan from Australian Clinical Labs have written a compelling paper titled The True Impact of Australia's COVID-19 Lockdowns on Critical Health Diagnosis. In this paper, they analysed the delays to medical appointments, cancer screenings and pathology tests of chronic health conditions over the past 5 years. And the results are truly frightening. To give you some frame on this, cancer diagnoses across the country, and especially Victoria, are down 18.9%. But what does that mean? 1,000 Victorians were not diagnosed with melanoma in the last period. 650 breast cancers and 600 bowel and 620 prostate cancers go undiagnosed. 140 lung cancers are undiagnosed. These are absolutely staggering omissions. Looking at the graph on the right-hand side showing the critical difference between New South Wales and Victoria. 16% of diabetic patients have gone undiagnosed and untreated in the past 12 months. To put that into perspective, 1% equates to 17,000 tests. 17% downturn in cholesterol testing and delays in treatment to or unmanaged conditions have occurred over this period. However, there were some interesting upsides to the pandemic that occurred. Prenatal testing is up 200%. We will have quite a number of COVID-19 babies on the way. Vet testing is actually up 300%. The purchase of COVID-19 puppies have gone through the roof. And finally, Zoom is now a thing. Our pathologists now easily dial into multidisciplinary meetings and provide expert advice remotely, adding to critical care and increasing their efficiency to talk about critical cases throughout the country. So I guess in summary, please never limit yourself to what you want to achieve in life. Never believe that you must follow the obvious path of your career and life. Make sure, however, to be an expert in something in your career that is valuable. Be that go-to person for something. For me, I learned how to transform big businesses that were struggling. I'm not a scientist, but I played to my strength and partnered with those that could support me on that journey. Believe you have an exemplary career ahead of you, and don't be afraid to take the path less travelled. It may take you to places that you never thought possible. Thank you for joining me today.
Thank you so much, Anthony. I really appreciate your presentation today, and certainly it exemplifies the roller coaster ride that you and your company had followed, and certainly dispels many of the myths that I guess would have been circulating in our community. So just for our participants today, we have really already received a large number of questions in the Q&A, but just reminding you where you can insert your questions. We have some minutes to respond to the questions, and I'll pose those to our panelists today. So I'm going to stop sharing now, and just for our panelists then, I'll start off by asking a question that came in pre today's session that came from one of the attendees. What are the challenges identifying the COVID virus, and how do you differentiate between COVID and other types of viruses? Either one of you, I'm happy for either one of you to respond.
Anthony, do you want to go ahead?
I was thinking that might be one for you.
Okay, so as I explained through the lecture, there are two possibilities, two strategies. One, you using the oligo test, you can detect in sequence. You know which virus it is. Second, you can recognize the structure of the protein, because each variants, when they mutating, they will also change the protein structure. So you can test the protein structure as well. And in terms of the rapid test, we're developing the detection to target both nuclear protein and spike protein. Spike protein is more specific to the COVID SARS-2. Okay, the nuclear protein is specific, broadly cover all the coronavirus, like the HIV and other coronavirus. So the spike protein is difficult to detect, but this is where the technology will go.
Thank you very much, Dayong. Yeah, so indeed, it's by testing the genetic material or the protein material at the surface. That's excellent. Thank you. And then maybe another question for you, Dayong, and then I'll go to some of the questions that our participants have lodged today. Will the tests such as yours be sufficiently affordable to be made available to the public through pharmacies, for example, for use at work and home?
We still don't recommend this test to be used at home because as long as we haven't proved the saliva test. The nasal swab, is not comfortable. I don't think everybody can use the stick properly in their nose, but the cost of the technology is getting down and lower. And so the cartridge on my hand is the ones produced by Alkaliza in Perth. So the cost of this cartridge only costs $1. Okay, the reagents on this cartridge it only costs a few dollars. So the overall cost could be between $10 to $20 at the customer hand. So potentially this test can be a few dollar test, but depends how sophisticated the government approval and how much more data will be needed, and that's all will build into the cost of the technology development.
Yeah, indeed. And I know from our own association, Dayong, that you do try to make tests that are called point of care tests. In other words, you make them really easy to use for general public, but of course within a limited parameter as specified for their use. All right, so I'm now going to go to a question that came through the online chat. And again, for either of you, is the original or alpha strain of the COVID virus still prevalent, or does each strain become superseded by the next one that comes in?
It's a good question. I would say when we're testing, they definitely look at the genetic material to see the thread. So in our lab, the predominant what we'd be looking for and the volumes that we're looking for would be the Delta strain. So the actual strain is probably immaterial to the time getting back to getting a negative or a positive result. But there's definitely a high proportion of that genetic link.
Yeah, indeed. And I do recall from the previous session that we had on COVID, so just for our participants today, this is the second in the series, and I invite you to visit the UTS Science website for the first in the series, where we had a member from New South Wales Health Pathology as one of our panelists, and they indicated that the predominant strain that is currently being detected is in fact the Delta strain. All right, so another question that has come through is effectively, will we have to really learn to live with the coronavirus into the future, or will it be suppressed?
I can give you a pathologist view. So
Go ahead.
I was sitting on a pathologist meeting today, and they were walking through, and pathologists are very balanced individuals, and they were saying that this will be here for quite some time. They talked about in what forums, but their belief is that if we are thinking running an organisation that we are looking for that this will move on and it will be something in the past, they were saying that you need to bolster your organisation and bolster life, that this will be part of life.
Yeah, indeed. No, I've heard other experts make similar comments that we're going to have to learn to live with coronavirus for some time into the future. All right, so another question, and Dayong, you touched on this to some extent in your presentation. How effective is hand sanitizer, such as the type that's used in hospitals, in destroying the virus, and maybe in comparison to hand washing?
It's a similar effect. To my understanding, I'm not expert in this, to how to kill the virus. But recently, one of our postdoc member put together a review article and see how to physically, different ways to kill the virus. But hand sanitizer, because they have a high concentration of alcohol, they also, depart the virus. But soap is cheap and dirt and easy,
Yes,
so washing our hand. Yes,
indeed. And another one for you, Dayong, is when you were explaining the upconversion particles, you were explaining that they have the ability to absorb infrared light and to then emit visible light. That's why we can see it. So we shine a dark light on it, and we then have visible light. It's a little bit of a technical question, but is there a specific wavelength or band of infrared that we use or prefer to use?
Yeah, so at the moment, we prefer to use the bandwidth around 980 nanometer, around one micron. So 1 micron near infrared, and you can check the green in 550 and also the blue in 470. You can also generate the red color around 660. So they're very colorful, materials been discovered and demonstrated in the past couple of years. It's very interesting science, scientific material, and that's actually the whole team of my research group get addicted to this area for the last few years.
Yeah, I can see why that would be the case. Anthony, I don't know if you would be able to answer this one. I hope that you can. What exactly does it take to get a clinical test approved by the TGA or other branches of the government?
Yeah, there's a set of working up of the test. So you sort of got to get the technology within the lab right, and then a set of tests. You then get what they call reference material from a governing body. That reference material will be a positive, so to speak. So the biggest issue at the start of the pandemic is that you could not get positive material. So remember at the start of the pandemic, we had very few positives. Indeed. And so it was very difficult. And they were basically synthesizing and trying to make it, but we would have paid thousands just to get a positive. So we could actually, so when you get those positives, you actually then have to work up the test and correlate that to the positive. And as soon as you're within a set of bands and you run a predominant sample size, there is a governing body called NATA within this country that actually will then certify not only the equipment, but the testing process that you've got. And then that can then be broadened to other types of, so every time you introduce a new piece of equipment, you need to re-go through that process. That's very rigorous in this country.
Yeah, indeed. I certainly believe that that's true. And I understand that the issues would have been relatively difficult at the beginning, difficult to manage at the beginning, when indeed you had no access to particular specimens.
Material, that's right.
Yeah, so to our panelists today, thank you both so much for your presentations. I really value the insights that you've brought to us. And also to our audience today, thank you so much for joining us. I hope that you have enjoyed the presentations, and thank you for staying with us today. Enjoy the rest of your day.